Vanakkam. Welcome to this video on our course Classics in Neuroscience. In this video, we start our discussion on a new topic which is on genes and DNA. This forms the second chapter of our book, Genes starting with the DNA. In this video, we will be focusing our attention on three topics: historical depth of molecular biology, genes and their terminology, and the clues to the DNA. The discovery of DNA was perhaps one of the greatest discoveries ever made by humans, which made the decade of 1950s unarguably the greatest, or inarguably the greatest decade in the history of science, in the history of biology definitely, in the history of science perhaps arguably. This discussion also involves, or also must involve, some discussion of competition that existed between labs and the ethics of competition that existed between labs, and how recognition may or may not have been given to women researchers who properly deserve to be recognized for their contribution. We will discuss this in some greater detail, but this is also part of our discussion. Now, in the 21st century, we take how the replication of information happens and how information is processed within the cell. For example, the DNA, RNA conversations. These are things that are now taken for granted, but until a century ago or even later, until 1950s, many of these things were very hazy. But the building blocks for this work were done not in the 20th century, but rather in the mid 19th century. In the mid 19th century, scientists proposed that a molecular explanation for replication of information, replication of cells will become available soon. This was a distant, vague idea, perhaps even can be called as an irrational idea, but it was a dream. This is why it is important to dream about things that never exist. In the 1850s, when this idea was first proposed that there will be a molecular explanation, it was weird and vague and completely unacceptable with the current state of reality. So they dreamed about things that never existed. So they said that, if and when we have this molecular explanation, the golden era of molecular biology will start, and that actually happened, except that it took 100 years for this dream to be realized. For example, histology. In histology, when the first textbook came in the 1850s, it said the molecular components of the cell and the inner constituents of the cell, the plasma membrane and the nucleus, when all of them are discovered, the golden era of histology will begin. This actually became a reality in the 1950s, but to think that someone was actually dreaming about this 100 years prior is, unbelievable today to think about. So it is important to not only work on real problems, but also think about problems or think about challenges that never exist, or think about a reality that does not exist today, which is why they say imagination is more powerful than knowledge. Remember, science is a creative pursuit. You need to think about things that do not exist in the present day for you to realize them into existence in a future date. In the 1850s textbook on histology, Kolliker also mentioned a law of genesis, a law of genesis. Something that is unthinkable. Of course, remember the manner and the meaning in which genesis and genes are being used today is different from the manner in which Kolliker meant it in the 1850s. So, there is a context to this, but still that someone could mention this as early as 1850s is a testament to the creative thought process that science is. As young students of the discipline, we need to keep this in mind. We need to stay in reality to solve problems using existing methodology or come up with new methodology, but it is also important to think about what the future holds, right? Be creative. Also, the phrase cell genesis is often used in a context that is different from what it is now, but this is perhaps what set the stage for the word gene to come about. Genesis has its roots in the word gen, and the word gen in Greek means to produce. This root, for example, is also shared by words such as generate, for example. Generate means to produce, for example, right. So, how cells originate, how are they produced, we need to understand this. So, for that we use the word or the phrase cell genesis. It is from this phrase the word genes later came about. The adjectival form of the word genesis is genetic. Now we are speaking about something, because this is a phrase that we see quite often: genetic origins, genetic engineering. This is a phrase that we see, this is a word and a phrase that we hear quite often nowadays. But in the 1850s and the late 1800s, it was used in a slightly different context. For example, even as early as Darwin, this word was used, but in a different context. For example, while describing the physiology of the spinal cord, the textbook of physiology by Foster speaks about the genetic component of the spinal cord. There, what is meant is rather what we would today call as, in the good old days, it was meant in the context of developmental, used by Foster's physiology. This is 1890s. Right. Darwin, in his Origin of Species, speaks about genetic connections. What he means by that is genetic connections is common origins. Now this makes some sense that when you speak about genetic connections, you are speaking about common origins. Later, people became more interested in understanding heredity. The first steps or the rudimentary steps that built our current understanding of genetics started with the chromosome theory of inheritance. When people put cells under a microscope and they observed, they found bodies with different colors. Chromo means color, some means body. Also you can relate these roots. For example, when you are speaking about other words that use chrome, for example, chromatography, for example, photochrome, for example, and when you are speaking about some, for example, soma or somatic refers to the body, for example. So when you say chromosome, what you are speaking about is colored bodies or bodies that appeared in various colors. So, this was observed as early as 1870s, 1880s, when people were examining cells under a microscope. Also, around the same time, mitosis and meiosis, as we now know, were described. That is, mitosis as it occurs in the somatic cells, that is a division of a cell into two daughter cells with different chromosomal pairs, and meiosis where the two daughter cells have the same chromosomal pairs, right? Cell division that happens with the same chromosomal pairs. These are things that we know from high school biology. These were described as early as 1880s, 1890s, around that time. Independently, Mendel also studied green peas and how heredity was observed in green peas. Remember, this is different from the studies performed by the histologists, which were mostly on animal cells. Green peas, being a plant and something that grows in the garden, was studied in great detail by Mendel. But somehow, for a long time, the conversation did not happen between these two fields. Long time as in about 30 years. After that, when this was discovered, this information was integrated into knowledge of heredity, inheritance and genetic science. So, Mendel's studies were from 1870s to 1900. Only in 1900 this work was discovered, or rather rediscovered, by the scientists who were working on genetics or heredity and inheritance. It was interesting because Mendel proposed that there were independent factors that contributed to features of heredity. In 1906, William Bateson, it is written as Williams, other is William Bateson, in 1906 defined genetics as the field of study of the rules of heredity. This happened at around 1906. Okay. Remember, Mendel proposed the phrase independent factors as the contributors of heredity. These independent factors were then called as genes. These independent factors came to be called as genes. So, genes means these independent factors. Genotype refers to features due to heredity or due to the genes or due to genetics. Phenotype refers to the combination of genetics as well as the environment. Genetics, when it interacts with environment. In the 1900s, 1910s, people started studying this fruit fly, Drosophila melanogaster. We spoke about model systems. Why this model system? What's specifically interesting about this model system is that it had rapid reproductive cycles. That is, it reproduces relatively quickly so that you can test hypotheses related to heredity and inheritance in the next generations pretty quickly, right. Well, actually whether that is rapid for today's time is something that we need to discuss, but in those days this was a excellent model system for study of genetics and inheritance. And around 1915 is when this chromosome theory of inheritance was proposed by Morgan. And then in the 1910s and 1920s, people started proposing this idea that there may be some errors in genetics that may lead to diseases. This was unheard of previously, that errors in genetics or errors due to heredity or inheritance could cause some metabolic dysfunction or some diseases. Right. This idea was later formalized in the idea of mutations. Mutations refers to, essentially the word mutation refers to change. In this case, a change that leads to a disease, change in genetics that leads to disease. People found that mutations could be induced by exposure to X-rays, also that mutations could be induced by exposure to chemicals. These ideas led to the development of induced mutations. How mutations can be induced and their effects can be seen in future generations of the organisms. In the 1930s and in 1940s, people started studying these principles or checking these principles using microorganisms, using bacteria. The advantage with bacteria was that they really had rapid reproductive cycle, even more than, for example, the fruit fly, right. So, again the same idea, rapid reproductive cycles that you see in fruit fly, much more rapid than perhaps even the fruit fly. So, microorganisms became the model systems or the preferred model system for this kind of studies, and people started observing that even organisms, microorganisms such as bacteria were becoming infected by other organisms which they did not know what it was at that time, that killed or that ate these bacteria. These were called as bacteriophages. Of course, we now know that these bacteriophages are actually viruses. Right. In fact, at one time it was thought that we could use virus as a means to reduce infection, as a means to treat infection, because virus can kill bacteria. Of course, with the discovery of penicillin and antibiotics, this idea was then left alone perhaps, but this idea that there were these bacteriophages that were affecting bacteria themselves was observed at around the same time. We are talking about 1940s, 1950s, 1930s to 1950s, that idea, that time. Also in 1946, it was observed that genetic information can be exchanged between different strains of mutant bacteria. This was the first example of what is now called as genetic recombination. It was shown by Tatum and Lederberg in 1946, where it is possible for mutant bacteria to exchange information to produce new information, right. So, genetic recombination. In the 1930s and in the 1950s, 1930s to 1940s to 1950s, physicists became interested in explaining how this genetics could work. Doyens of physics, Niels Bohr and Erwin Schrödinger. Interesting that these are doyens of physics who asked questions about the fundamentals of biology, a field in which they had little to no experience, but they asked some crucial questions, answering which led to some explosive growth in the field of biology. They themselves may or may not have, it is debatable about their contribution to this field, but they asked the right questions. Niels Bohr asked this, suggested that there is this complementarity between these independent factors, right, that can explain the physical nature of life, for example. Erwin Schrödinger in 1940s wrote a book on what is life, in which he asked questions about the hereditary nature of biology. Of course, there were some errors that are now apparent in the biology part of the material. However, the questions that he posed were interesting enough, aroused not just the interest of the scientists, but also the interest of the public into understanding what is indeed life. These questions raised enough interest to, or enough curiosity among scientists, that later led to the development of the field of molecular biology as we now know it. Arguably, you can say that the discovery of bacteriophages or viruses as the beginning of the field of molecular biology as we now know it. Okay. Although the structure of the DNA itself was discovered much later, was described much later, the idea that genetic transformation can happen was first discovered in 1928 by Frederick Griffith, where he observed this in pneumococcus bacteria, genetic transformation between two different strains of pneumococcus bacteria. In 1943, description of a transforming principle of this heredity, transforming principle. In 1943, MacLeod and McCarthy described the transforming principle as a long chain nucleic acid, also called as deoxyribonucleic acid, which we now know as DNA. Many people think wrongly that this 1943 study was unnoticed until Crick and Watson discovered the structure of the DNA. This is not true. Actually, it is quite well known. It was well known and recognized in the 1940s and in the 1950s that there was this idea that the transforming principle was indeed the DNA. This was known at least in the field of molecular biology. People who are working in the field knew that this study. Now, common people think that no one knew about DNA until Crick and Watson discovered. This is not true. People did know about DNA. People who work in the field did know about DNA 10 years prior to the discovery of the structure of the DNA. In 1952, Hershey and Chase suggested that genes are indeed what we call as DNA. So, DNA is the gene, or this was suggested, but the elusive material until that time was what is the structure of DNA, and it was believed strongly by that time that the structure of the DNA will contain the clues to our understanding of heredity and inheritance, how heredity works. That is something that we can understand if we understand the structure of the DNA, was very strongly believed by the early 1950s, 1950 to 1953. By that time, the hot question was the structure of the DNA. Everybody wanted to understand the structure of the DNA, or those who are working in this field were pretty keen on discovering the structure of the DNA. So, there is race to identifying the structure of the DNA by 1950s. With this, we come to the end of this video. In this video, we discussed history of molecular biology, genes and terminologies related to genetics, and clues regarding the DNA. Thank you very much for your attention.